detected for the first time in Brazil on the coast of the southeast region in the late 1980s, when oil and gas prospecting began in the Campos Basin offshore of Rio de Janeiro. Sun corals of the genus Tubastraea are now spreading very swiftly throughout the rocky shores and cliffs of Brazilian islands and are considered to be biological invaders. Once biodiverse and multicolored, the reefs in Búzios Island, part of the municipality of Ilhabela, Sao Paulo State, also located at the southeast, are now covered with orange stripes. In some places, no bare rock or other species of coral can be seen. The reefs around Búzios Island are in an irreparable condition, said Marcelo Kitahara, a professor in the Marine Science Department of the Federal University of Sao Paulo, DCMARUNIFESP, in Santos, Brazil. Kitahara heads a project supported by the Sao Paulo Research Foundation, FAPESP, to study the phylogenomics of two species and the links between their evolution and climate change. The genus Tubastraea comprises 7 species, all native to the tropical waters of the Indian Ocean and Pacific. Only 2, T. coccinea and T. tagasensis, are also found in the southwest Atlantic. Both are invasive species. First Brazilian sightings were recorded in the Campos Basin in the 1980s, followed by the discovery of colonies on reefs off the southern coast of Rio de Janeiro state in the 1990s. Since then, sun coral has been found across over 3,000 kilometers of the Brazilian coastline, from Santa Catarina in the south to Ceará in the northeast. Management action is still possible in some places, but this requires the complete manual removal of all colonies, Kitahara said. If nothing is done to stop its advance, sun coral could potentially colonize the entire Brazilian coast. A study showing the surprising capacity of sun coral to regenerate had its results published in the Journal of Experimental Marine Biology and Ecology. The lead author is PhD student Bruna Luiz Pereira Luz, a biologist affiliated with the Federal University of Paraná, UFPR, and currently in Australia studying sun coral at James Cook University, Townsville, near the Great Barrier Reef, under supervision of Kitahara. Sun coral colonies multiply at a great speed in areas such as these. We set out to understand how and why, said Kitahara. On one of the findings, only made possible through a lab-conducted experiment, they also revealed that sun coral regeneration process gets faster as water temperature increases. Possible influence of oil. The Gulf of Mexico also has vast offshore oil fields, and sun coral has been found on the Mexican coast since the early 2000s. There are even records of sun coral attached to the hulls of ships. We can't be totally sure that offshore oil drilling in the Campos Basin resulted in the invasion of our coast by sun coral, but all the evidence points to this conclusion, said the FAPESP project coordinator. A coral reef is a limestone skeleton built by colonies of thousands of tiny animals called coral polyps. Most reef-building corals contain photosynthetic algae that live in their tissues. The corals and algae have a symbiotic relationship. The polyps provide the compounds required by the algae for photosynthesis, and the algae provide the polyps with nutrients. Other types, including sun coral, can grow and proliferate without algae. Because it has no algae, sun coral isn't confined to places with sunlight for photosynthesis. It typically occurs at depths of up to 20 meters, but sightings have been recorded at 110 meters. On rocky shores and underwater cliffs, polyps build huge numbers of colonies and cover 100% of the substrate, Kitahara explained. During this process, they drive out the native coral, devastating ecological relations with the marine fauna that depend on or inhabit it. Reorganization of stem cells to investigate the mechanisms that enable sun coral to adapt so successfully and proliferate rapidly in various marine environments. The researchers collected a colony of T. coccinea and another of T. tagasensis from Búzios Island. In the laboratory, the researchers removed from each colony 120 fragments composed of skeleton with living tissue but lacking mesenteries, mouths and tentacles. The samples of each species were then separated into two groups of 60, one with very small fragments, 3.5 to 11 square millimeters, and another with slightly larger fragments, 11 to 53 square millimeters. All 240 fragments were placed separately in containers with filtered seawater. For each combination of species and fragment size, individuals were further separated into three groups of 20 fragments and maintained at a constant temperature of 24 degrees Celsius. Historically, the average surface water temperature in the region, 27 degrees Celsius, the average sea surface temperature in summer, or 30 degrees Celsius, observed during heat waves. Finally, the effects of the presence of food were tested by adding equal amounts 10 milliliters of live zooplankton every other day to half of the containers. The fragments were photographed on the first day of the experiment and when the mouth and complete polyp were first observed. Only 41 of the 240 fragments, or 17.1%, underwent tissue necrosis and death. The other 199 fragments, 86.9%, regenerated. Of these, 21.9% of the entire sample displayed an alternative regeneration pattern, with the formation of two polyps instead of one. Regardless of the species, coral fragment survival was affected only by temperature. The survival rate was highest at 24 degrees Celsius. There was no difference between the fragments kept at 27 degrees Celsius and those kept at 30 degrees Celsius. Food supply and fragment size did not affect survival. Regeneration was found to include the following stages. After initial tissue retraction, mouth rudiments became noticeable, sometimes two for a single fragment. Subsequent development consisted either of tissue reorganization around the mouth rudiments, leading to the formation of two small, distinct polyps, or the reabsorption of one of the rudiments, in which case significant tissue differentiation around the remaining mouth rudiment resulted in a larger polyp. We observed a very interesting phenomenon, Kitahara said. From a cellular viewpoint, there was a reorganization of stem cells. The polyp in formation consumed tissue as a source of energy to prioritize the production of other body parts. The results of the experiment generally indicated faster regeneration rates at higher temperatures. The fastest mouth regeneration for fragments without contact with food was 23 days at 24 degrees Celsius or 18 days at 30 degrees Celsius. However, fragments kept at 27 degrees Celsius in contact with living zooplankton displayed 30% faster mouth development. This suggests optimal mouth development at the intermediate temperature 27 degrees Celsius, provided there is contact with food. Fragments of both species developed into complete polyps in approximately 25 days at 27 degrees Celsius and 30 degrees Celsius. Unfed individuals of the species T. coccinea took approximately 41 days to achieve polyp formation. As Kitahara explained, the fact that sun coral regenerates faster at higher temperatures is highly germane to its invasive success. Most native corals on the Brazilian coast suffer bleaching when surface water temperatures rise. They lose color, said the FAPESP-funded researcher. Warmer water interferes with the metabolism of their symbiont algae. Bleached coral survives only for a few days. If the temperature remains high, it dies. The bleaching or death of native coral opens up an opportunity for substrate invasion by sun coral. The next steps in this line of research, according to Kitahara, will be to sequence sun coral's genome, on the molecular side, and, on the ecological side, to investigate the biological aspects of its invasion and how it affects native marine fauna. The future does not appear promising for native corals on the Brazilian coast, in Kitahara's opinion. For sun coral, on the contrary, it appears to be bright. On one hand, global climate change and rising seawater temperatures help the invader, which regenerates faster in warmer water, while native corals risk dying. Not to mention the prospect of expanding oil production in Brazilian waters. FAPESP is aware that the very best research can only be done by working with the best researchers internationally. Therefore, it has established partnerships with funding agencies, higher education, private companies, and research organizations in other countries known for the quality of their research and has been encouraging scientists funded by its grants to further develop their international collaboration. For more information, http://www.fapesp.br/n